Good evening, everyone, and welcome to Broken Brains, the online Pint of Science event brought to you from Manchester. My name is Jill, and I'm one of the coordinators for Pint of Science, and I'm also a PhD student studying neuroscience here at the University of Manchester. I'll be your host for this evening, taking you through the eclectic mix of short talks that we have planned for tonight, all centred around my personal favourite area of research, the brain. Essentially, we're bringing you 3 shows in one tonight because we're covering such a broad range of topics, so hopefully there's something for everyone. We'll be taking a look at how things work and what happens when they don't. Please go ahead and use that chat function to send in your comments and questions throughout the show, as we'll be keeping an eye on this and trying to answer as many as possible. After each talk, there will be a short Q&A, so please have your questions ready for then. This is the last night of our 4-day annual festival celebrating science, and we want to know which shows you've been to so far this week, so let us know in the comments. These events wouldn't be the same without interaction from the audience, so to kick off the discussion, I've got a bit of trivia for you all. Try and guess the total length of blood vessels within the brain. See how close you can get without looking up the answer online, and we'll come back to your answers later in the show. First, let's see where some of you are joining us from tonight. So we've got one person from Manchester. Is anyone else joining us from anywhere further than Manchester? Portugal, that is a bit further, definitely. Anyone else? Another one from Manchester, keeping it close to home, that's great. So it is good to see that people are joining us from locations outside of Manchester and even outside of the UK. It's definitely one of the perks of doing these events online. Now, I hope that everyone has a pint in hand or a cup of tea or a glass of squash, whatever it may be, drink up, sit back and enjoy the show. Now, our first speaker of the night, Jayesha, is a PhD student like myself in the Division of Neuroscience and Experimental Psychology, and her talk will cover how the brain processes sign language compared to speech and what this means for brain organisation. Jayesha, it's great to have you here tonight. Over to you.
Thanks, Jill. Okay, so language is how we communicate with other people. We learn and use words in a structured way. I grew up speaking multiple languages, Gujarati at home and English at school, and I always loved learning them and learning about the ways that they're used in our society. So I think in some ways it was inevitable that I would end up researching something related to language. So for most of us, language is synonymous with speaking. And actually, it wasn't until I was looking for a topic for my PhD research that I was exposed to sign language research. And this is what I'm going to share with you today. So for my PhD, I'm designing experiments to look at how the human brain is involved in making sense of sign languages and how these might be different from spoken languages. So here's a picture of a slice of my brain. This is taken in an MRI scanner. So some of you may have seen these in hospitals or even been in one of these before. So what we do is use these for research, and you lay in a scanner and the MRI takes a 4D image of your brain. So we'll go back to this picture now. So you can see here, this outer bit here is the skull. These squiggly bits are where you process some of the information that you get from your senses. And the brain is split into 2 parts. You've got your left side and your right side. And this white bit here connects the 2 and allows those 2 parts of the brain to communicate. So in my talk today, I'm going to first of all go through some of the similarities in the linguistic features of speech and sign, and then how these manifest in the brain. And then lastly, because this is the Broken Brains Pint of Science, what happens when language goes wrong. So to start off with, the fundamental differences between speech and sign are quite obvious. Speech is sound-based, whereas sign is action-based. There are more than 6,500 spoken languages in the world and about 300, maybe more, verified sign languages in the world. Sign languages develop in deaf communities where there are needs for the languages. And they've only been recognized recently. So it could be the case that many sign languages exist that are not verified yet. So I'll give you an example of a sign. So this is taken from the UCL Sign Bank, and it's this BSL sign for, BSL is the British Sign Language sign for science. BSL is the most commonly used sign language in the UK, And just as with spoken languages, there are regional dialects and variations in the way that language is used across different cities in the UK. So I can give you an example of how this can be explained in speech. So the ongoing bread debate, I'm from the East Midlands, so I will always and only call this a cob. But I know that many of my friends refuse to do that and will call it something else. I'm not sure what they're called in Portugal or in Ireland, I'm sorry. But in a similar way, there are these kind of differences in sign language. So here are two signs for mother. The first one, that's a sign that I learned learning BSL in Manchester. And the second one, I believe, is the sign more commonly used in London. Right, so there are these differences, but also they're quite similar in the fact that these rules exist in both of the language types. And a further rule that exists is grammar. So both language types have a unique grammar, and just like two different spoken languages have rules, sign languages have the same rules. So, for example, all languages have a word order. So in English, I would say, my name is Jayesha. In Hindi or other South Asian languages, the word order changes. So my name, Jayesha, is the way that the language would say that sentence. In BSL, that is different again. You'd say, name me, Jayesha. So how about these things in the brain then? So what happens in the brain? There are two important things to consider when we think about what happens in the brain. We want to understand what somebody is saying to us, and we want to be able to speak back at them. So I'll talk about language in the brain in relation to speech first, and then sign later on. So this is the left side of the brain. In most people, language is processed on the left side of the brain. So what happens then? So when somebody speaks to you, the sound information that runs through the air reaches your ears and then moves on to what we call the auditory cortex. This is where sound information that's language related is separated from just background noise that's not language related at all. And in this area here then, including what we call Wernicke's area, these sounds are made sense of as language. There's a direct link between this area and what we call Broca's area, which is important for coordinating different areas in the front of the brain that allow you to actually speak. So this Broca's area will send a message to the motor cortex, which will then send a signal to your muscle so you say the right things. So here we go, auditory to Wernicke's, to Broca's, to the motor cortex. And the motor cortex is quite easy to find if you want to on your own brain. So if you find the middle point at the top of your brain and then slowly work down to the corner of your left eye, that there is what we call the motor strip. And it's organized by body parts. So you can see the lips here. These are what we'd use, the lips and the tongue. Here's what we'd use if we were speaking. And then here are the hands, which are what we use when we're signing. So let's go back to this image here. This is the same one that we've just seen, but when we talk about sign languages, there's something missing, and that's vision. So sign language is a very visual language. So it would make sense then that more visual areas of the brain, so these are at the back of the brain, are involved in processing them. And we can see these things in brain scans. So brain scanning, like I mentioned at the beginning, allow us to look at pictures of the brain, but we can also see the specific areas of the brain that are involved, comparing sign language and comparing spoken language. So in the MRI, people lay down and we ask them to do a task. And while they're doing the task, what the MRI is taking a picture of is the blood flow that is flowing to the areas of the brain that are being used during that task. So these are what these yellow bits are. So in this task, deaf people that knew sign languages from birth were asked to watch sentences in BSL. And hearing people that knew spoken English from birth watch and listen to somebody speaking. So these yellow areas are the areas that are used during these tasks, and and as you can see, there are very similar patterns of activation in these areas. So here are the visual areas that we mentioned that we'd see, which is expected because they're more visual, the sign languages are more visual languages, but you can also see that there are some similarities across these two parts, and in similarities and patterns even on the right side of the brain. So it's interesting to see that even though these deaf people don't have any sound input to their brains, show activation in areas that are important for processing sounds. So this just really tells us that even though the areas of the brain are for speech, it doesn't mean that speech is the only thing they can process. So these areas in deaf people, with the absence of sound information, are now being used for something different, are now being used for sign language in a different way that hearing speakers use those areas to process speech. We'll go back to this picture again. So now we know that the brain areas that are involved in these language types, we can see, we know what to look out for when things go wrong. So if there is damage to these understanding areas of the brain or to these areas of the brain that are important for coordinating movement, you can expect to see a problem with the language output or understanding the language. So I'll show you what that could look like on a brain scan. So again, this outer bit is the skull. This is these are the squiggly bits. This is a picture of right down the middle of the horizontally across your face, and this here you can see is a gap where there's been a lesion caused by a stroke. You'll hear about stroke, what stroke is properly in the next talk, but the damage caused by stroke can affect patients in multiple ways depending on where that lesion is. So to test what's actually happening with language function, patients are often given a task. So you can see this picture and they're asked to describe what the picture is. And people with damage to those motor areas are not able to get the words out, even though they know what they want to say. People with damage to the understanding areas of the brain can just talk and talk and talk, but the words that they say don't make any sense at all. A similar thing happens in sign languages. So patients are not able to produce the signs needed, or they're not able to understand the signs, depending on where the damage is. So we're coming to the end of the talk now, and I just want to leave you with 3 key messages. So speech and sign are used differently, but they have comparable linguistic features, and they both show similar brain activity. Studying both of them can help us to find areas of the brain that are important for language, that are core regions for language, not for speech and not for sign. And this allows us to gain an enhanced understanding of the healthy brain, which then lets us have a baseline to come back to when things go wrong. So when patients are undergoing therapy, if they've had damage caused by various things, we know what the brain wants to look like. We know how the brain wants to function on a normal day. Thank you very much for listening. And if you have any questions, I'm happy to answer them.
Thank you, Jayesha. I had no idea that there were so many signed languages in the world, or that sign languages had different dialects. Yeah, I really enjoyed that talk. Hopefully we have some questions. So if anyone does have any questions for Jayesha, please do type them in the comments section and we'll bring them up on screen. So if we have any, please can we have those on screen? So first question from Rose is, do you think therapy and basic science should work collaboratively more to produce better treatments?
Absolutely. I think it's really important for the reason that we do basic science is to be able to give something to the community, to people that we do the science for, right? So I think that collaboration should be the key to how we do all science and research and therapy, personally.
Definitely. Okay, hopefully you have some more as well. Okay, we have got a question from Phil. From Megan. Do you know what the brains of people who can neither hear nor speak look like, as in those that use touch for communication?
Do I know what? Yeah, I guess so. People that use touch for communication, they use Braille, don't they? I personally don't I've never looked at this in the brain, but I can imagine that if they don't have sound input and they don't have language input in that way, those areas of the brain that you or I would use for speech are taken over by the areas that are more responsible for movement. So there's greater activity in those areas. And I showed you before the motor strip with the hand. So those body parts are organized not only by where they are in the body, but the size of those body parts is dependent on how much a body part is used. So the hands are quite big because there's loads of nerves in the hands, so we use them often. So I imagine somebody that uses touch for communication will have larger surfaces of those kind of areas in the brain.
Right, yeah, that does make sense. Similar to like the homunculus with the representation of nerve endings.
Yeah, exactly.
So next question. Question from Phil. As a kid, I had a mystery illness, potentially a viral infection to my CNS. I recovered but had developed speech impairments, dyslexia, and more. Can viruses affect brain activity or speech?
I think that depending on where that virus reached your brain, yeah, absolutely, it can affect brain activity and speech. But also, like you mentioned, you recovered from that. So the brain can be damaged, but there's always, because of the way that the brain is organized, the brain can get better. But it depends on where the damage really was.
Mm. Yeah, definitely. So next question we have is from Mizzy Peasy. So if a person has a speech impediment, can it affect the signing in any way?
Okay, so if someone has a speech impediment in a spoken language, that might, if someone has speech impediment in a spoken language and they don't know sign language, then I don't think so. But if someone has a sign impediment, that can affect how they sign. So they can have an impediment in how they sign. But yeah, does that make sense?
So a sign impediment, is that something specific to signing, or is that just like a general motor impediment?
I think so. I think it's specific to signing. So a motor impediment would mean that you can't move your hands, right? But an impediment that's specific to sign language would mean that you can't produce the language for the signs. So people that have damaged language areas of the brain that are signers only, they are still able to produce gesture sometimes, but they can't produce the signs.
Right.
For language. Yeah.
So I think we do have a couple more questions. So the next one is from Priya. How developed is the baseline at the moment? Is it something still being researched?
So I assume this is the baseline about the healthy brain. So I think I only research the healthy brain. I don't do any research into damaged brains. So for me to have a job and for people like me to be able to do, basic science means that it's because there's always something to be researched and there's always something more to discover. So the more and more we learn about sign languages, the more and more we can understand that baseline better. And because they're relatively new languages or recognized relatively recently, it means that there's a lot to learn. The strength of sign language research compared to spoken language research, they're completely opposite. So there's still a lot to do for the sign language baseline. Yeah.
Cool, thank you. I think we do still have some more. So next one from Andrea. Do you think speech is developed by nature or nurture?
I think that we all have an inherent ability to speak, but the way that we speak and what we say is developed by nurture. So there are some studies that show that babies before they start to speak the language that they're of their countries or whatever is spoken in their household, they're able to distinguish between different sounds in different languages, which they're not able to do later on. So it shows that there's an inherent ability to learn any language, but what language you then are made to speak or are brought up to speak is the one that you develop further.
That's incredible. And I think I heard somewhere as well that babies, similar to babies that speak, they babble in sign language. So as they're learning to sign, they kind of make gestures that are similar to sign language, but not quite sign language.
Yeah, so I think that deaf children who learn sign language from birth, they have the same developmental milestones that hearing children have. So you'd babble, and you'd learn simple signs and complex signs, and you'd move on from there. But I'm not a sign language expert. I only know the basics. So if you want to know more about that, you just go to a deaf person.
Definitely an expert compared to me, but yeah, thank you so much, Jayesha. I think that's all we have time for right now, so that's the end of our Q&A. Thank you again for that talk. It was really interesting. Now, moving swiftly on to our next speaker, who is our very own President and Vice Chancellor of the university, Nancy Rothwell. So Nancy, it's a privilege to have you here to demonstrate your knowledge of neuroinflammation, which I don't think that many people are actually aware of. Nancy, in fact, founded the Brain Inflammation Group, which is the lab that I currently work in. So Nancy will be talking about inflammation in stroke. And without further ado, over to you, Nancy.
So thanks very much indeed. It's great to be here, and having heard Jayesha talk so well about what happens to your brain when it's damaged through a stroke, I'm going to tell you a little bit about stroke. So stroke happens when there's not enough blood flow and oxygen to part or parts of your brain, and those parts are then damaged. Sometimes they die. It can be cause death of the patient, and it's actually the third greatest killer in the Western world. And it's caused when an artery supplying the brain blocks or bleeds, in poor general circulation, in heart failure, in drowning, and even in babies during or just after birth if they're deprived of oxygen, called birth asphyxia. So where are we on understanding stroke? And here's a picture that's a little bit like the one you saw, but it's actually a post-mortem sample of somebody who's had a stroke. It's a massive burden on society, and obviously a huge burden on those people who suffer a stroke and their carers. And one of the things about having a stroke is even those people who don't die very often suffer very badly. They have speech impediments, just as you've heard from Jayesha, but often they also have memory impairment, they have difficulty moving and walking, and they can change their character. And that's one of the things that is the most distressing for many people who have a stroke. There are very few treatments. We're quite good now at limiting the causes for stroke, of reducing blood pressure, blood glucose, blood cholesterol, advising people on healthier lifestyles. But once somebody's had a stroke, there are very few treatments we have. And unfortunately now there's little interest from pharmaceutical companies and very little government funding. And the reason for this is probably because there've been so many failures. There have been hundreds of clinical trials of new treatments in stroke, and virtually all of them have shown no benefit. A few of them have been harmful. So the big question we've been trying to ask is, why is it? Why have we done so badly? Have the scientists got it wrong? Did we do the wrong experiments, the wrong targets? Did the clinicians who I work with very closely get the wrong patients or do the wrong trials? And we think that a key reason, one of many reasons, is in fact that we've been trying to target the wrong processes. And so what we've been doing is trying to target a process you'll all have experienced, which happens only in disease, something that is not present at all in healthy tissues, and that is inflammation. So anybody who's had an infected finger or an inflamed knee or something like that will have suffered from inflammation. It's the redness and the swelling. It's our own body's defense against an infection. And it's common in arthritis and asthma and inflammatory bowel disease. But this was the front page of Time magazine reporting that we now know that inflammation is involved in lots of diseases, in cancer, in diabetes, in the furring up of blood vessels, which is often a cause of a stroke, in obesity, in skin diseases, and now in diseases of the brain. So we now think that inflammation, which is an important defense response, if it's too severe or it goes on too long or it's in the wrong place, is massively damaging. And it's the cause of many diseases that we now know. So there's a huge attempt to understand what causes inflammation. What are the molecules that make tissues inflamed? Can we stop them? And that's what we've been trying to do in research in Manchester. And a key culprit in this is a molecule, we'll call it IL-1 for short. And it's a molecule that causes inflammation. It's often called the master controller. It's something that causes a fever when it's produced in very small amounts in infection. And it's something also that can cause disease. So it's a big disease target. And we know it's produced rapidly in the brain after a stroke, in a brain injury, and in dementia. Unfortunately for us, there is a naturally occurring blocker of IL-1, something we all make actually, that stops IL-1 from working, and it's called IL-1 receptor antagonist. This is incredibly rare. We've never found in biology before a molecule that does nothing but selectively stops IL-1 from working. And we think it emerged in evolution to help to stop the detrimental effects of IL-1. Because remember, it's good in small amounts in the right place, but if it's prolonged or severe, it needs to be stopped because it will cause damage. And that's what we think the naturally occurring blocker does. So through many years of research, many PhD students, I've now supervised over 50 of them, we and others have found that there is rapid production of all the different forms of IL-1 and its naturally occurring blocker in the brain. And that production seems to correlate with the places, spatial, and with the time, the temporal pattern, that suggests it might contribute to injury. But that could be circumstantial. It could be secondary. It could have nothing to do with the injury. So two important approaches in science to understand whether something is causal is, first of all, you see what happens when it increases. Does the condition get worse? And then you see what happens when you stop it from working. Does the condition get better? And in experimental studies, we found that increasing IL-1, whether that's administered or because it's naturally increased, for example, in a fever and in an infection, brain injury is worse. And this may be one explanation why a lot of brain diseases get worse during infections. We know, for example, that in chronic conditions like dementia, like multiple sclerosis, infection and fever makes it worse. And certainly in the patients that we work with that have had a stroke, they get very much worse if they have an infection afterwards. That might be because they've got more IL-1. But most importantly, we found that blocking IL-1, either stopping it from being produced or stopping it from acting with the naturally occurring blocker, markedly reduces the extent of injury. And we found that through experimental studies, both on cells, on tissues, on animals, and as I'll mention in a moment, now in patients. So obviously, a really important question then is this of any clinical relevance? Could we use blocking IL-1 as a treatment for stroke and perhaps for other conditions? And here we have to work in patients. And here we use a lot of the same approaches that Jayesha used. We use brain imaging to look at what happened after a stroke. And now with advanced brain imaging, we can also do functional imaging. We can actually see what's happening in the brain after a stroke. And one of the things we see is a big increase in inflammation. So the questions that we're now trying to answer are, does inflammation, whether it's chronic or acute, i.e. long-term or short-term, cause a stroke? Do molecules like IL-1, called cytokines, contribute to the outcome after a stroke? In other words, do they make it worse? And obviously, most importantly, is blocking IL-1 helpful? Well, we've got some of these answers, but not quite all of them. One of the things we know, for example, that in older people who are at risk of a stroke, an infection, like an infection of the lungs, causes an increased risk by 3 times of getting a stroke or a heart attack. And by the way, heart attacks are a bit like strokes. It's the same thing that happens, a reduced blood supply. So when there is a risk already, then adding infection and inflammation to it increases the chances of a stroke. We know that in human patients, there's production of a lot of the inflammatory molecules that we see in experimental studies, and there's production of IL-1 very quickly. But obviously, the key question is the last one. Is stopping IL-1 from working or stopping it from being produced helpful or not? We've now undertaken 4 clinical studies in stroke in Manchester at Salford Royal Foundation Trust. And the first one, the small one, was what we call a phase 2 study. When you do patient studies, they're usually in 4 phases. The first one is in normal volunteers. Small doses, increase them, make sure it's safe. The second one is in patients, because something that might be safe in a healthy individual might not be helpful in a patient who's had a stroke, who's already very ill. The third phase is when you do a larger study, and phase 4 is really observation when treatments are out in the general population. And a good example of this would be the vaccines that we've all just seen. So it's not until you're seeing phase 4, you're seeing millions of people vaccinated, that you see rare events that might be damaging. Studies like these are conducted in what we call double-blinded, placebo-controlled, randomized. And what that means is none of the investigators know what the patient is getting. We have vials that are numbered. Some of them are placebo, nothing more than saline, and some contain the IL-1 blocker. And a distinct, separate center randomizes what each patient gets. And then we look at outcomes in early trials of safety and feasibility. Can we give the drug? Are there any unwanted side effects? And actually, what we saw was that inflammation does contribute to stroke. The first trials we did suggested that blocking IL-1 was extremely beneficial. It reduced a number of outcomes. And we also found that it had effects in the brain and outside the brain. Now, what we then went on to do is larger studies, and we've done studies now also in patients who've had brain damage due to bleeding in the brain hemorrhage, and we've also found beneficial effects there. So it does look as though the IL-1 blocker is a promising treatment. Now, all this was going well when we were in the middle of what's called a phase 3 trial, a final definitive trial to see if it was going to work. We'd got partway through it and COVID struck. And so the trial has been halted ever since. We're still hoping to restart it. But what I want to finish with is saying that I've spoken about stroke, but there are studies ongoing because IL-1 and inflammation might also be important in a whole range of brain diseases, whether they're short-term diseases such as caused by a stroke or injury, or epilepsy, infections, tumors, dementias, Parkinson's disease, and so on. So there's real hope that we might actually be able to at least reduce the damage caused by some of these diseases by blocking inflammation, possibly by blocking IL-1 or by others in addition to that. Thank you very much for listening.
Thank you, Nancy. That was really great and explained really well. I know that the topic of interleukin-1 can be quite a complicated one. It's amazing how many things can be reduced just by blocking interleukin-1, things like seizures. I wasn't aware of that. Shows the power of interleukin-1. So I think we have a few questions in the chat, if you wouldn't mind sticking around for them.
Yep.
Bring those up on screen. So question from Lucas, if interleukin-1 occurs when we have a fever to fight off an infection, are there any risks or potential side effects when it is inhibited?
So this was one of our worries. We're taking quite sick people and we're blocking IL-1, and we know that IL-1 is good at fighting infection. So we were really worried and watching very, very closely whether or not those patients that we treated had more or worse infections. Actually, we didn't see it, surprisingly to some extent. So it seems as though that's not the case. And it may be that other inflammatory molecules take over and then can do the job that IL-1 does in infection. So it is a potential risk. We think it might be more of a risk if IL-1 blocker is given repeatedly. For example, you might have to give it every day for years in dementia. And that the longer term studies are looking at whether there could be more infections. The last thing to say is that the blocker of IL-1 is in use already. It's used to treat people with rheumatism, and it's used in very rare genetic conditions in children who have a hyper-inflammatory response, huge inflammatory response. They've got permanent fevers and lots of problems, and they're given that IL-1 blocker every day, and it actually helps them to get a lot better.
Amazing. So it definitely has some potential. So we've got another question from Megan. Are you blocking interleukin-1 all over the body in these trials or just in the brain?
Yeah, really good question. At the moment, we're blocking it all over the body. Early studies, we thought about giving it directly into the brain. But in a stroke, you get inflammation, not just in the brain, but all over the body. You actually get what we call a defense response. So we think it's probably good to be blocking it everywhere. And one of the things we worried about was that IL-1 blocker is quite a big molecule. Would it get into the brain? And if it didn't, it might not work. But actually, it does get into the brain. But we do think that blocking it both in the brain and outside the brain is equally important.
Right. And we've got one last question from Mr. Blackstone. What sort of timescale is there between a stroke occurring and the IL-1 blockers being administered to see a benefit?
Really important questions. We think within 6 hours. Now, the only current treatment for stroke is something called TPA, which is what's called a clot buster. It breaks up the clot. The problem with TPA is if you haven't got a stroke because of a clot and it's because of blood vessels burst and bleed, it's absolutely the opposite of what you want. And that definitely has to be given early. The advantage of the IL-1 blocker is, unlike TPA, it doesn't seem to have any damaging effects if you give it later. So you don't have to have a CT scan before you get it. But we think that longer than 6 hours, there's too much damage already developed. And we think that probably it's better giving, the earlier the better, the quicker the better, but within 6 hours.
Really critical to get those acute time points. Okay, so that concludes the Q&A. Thank you so much, Nancy. It was great to have you on here. So before we move on to our final speaker of the night, let's reveal the answer to our trivia question. So in case you missed the start of the show, I asked the audience what they thought the total length of blood vessels in the brain is. So let's bring up some of our audience's guesses. So Nick from Southeast London is saying 100,000 kilometers of blood vessels. That's an amazing. Joe Merlini, my guess is 50 meters. So right down to the opposite end of the scale. Any other guesses? Anna is saying 5,000 kilometers. Yep, she's gone for the middle ground. Any more? I wonder. I think I saw some more guesses in there. 650 kilometers. Okay, I think that 650 kilometers might be the closest. Jacob says 6.3 kilometers. I'll tell you what the answer actually is. It is 600 kilometers of blood vessels just inside your brain. And I think that is incredible. It's actually one of the most vascularized organs in the human body. So I've got a second bit of trivia for you guys if you're feeling confident. And this is something that I actually only just learned last night in our Rise of Robots show. So if any of you were at that event, you might remember the answer to this one. So the question is, what is the total information content of the human brain in bits? And we'll come back to those answers at the end of the show after the next talk. So leave your comments, your guesses in the chat function. So now on to our final speaker, Emma. Emma is a professor of psychopharmacology, and she's joining us from Bristol to tell us why ketamine could be the next big thing in treating depression. Please welcome Professor Emma Robinson.
Thank you very much, and thank you for the opportunity to come and talk to you about our work. We're very excited that we've had the opportunity ourselves to get involved in this relatively new area, which relates to how we might use a completely different type of pharmacology to treat this very debilitating disorder, which is, of course, major depressive disorder. So I'm a basic scientist. Most of my research involves these guys. So I work with rodent models, but with the ambition that we will help to solve to provide new treatments for really what is one of the most challenging disorders, I think, that we face. And we all talk about how important our areas are, and we're all very passionate about them. But I think it's useful to just kind of put into context what we're seeing with major depressive disorder, which is that it is a really escalating problem. It's being described as an epidemic of the 21st century. It's really challenging because it's affecting young people particularly. And one of the concerns that really I have is that people often get this really quite young, and then it impacts on them for the whole of their life. So it's impacting. And the World Health Organization is suggesting that we're talking about 300 million people. 800,000 people are dying each year from suicide. And it's also not isolated to a purely psychiatric disorder. It's also very commonly associated with other things. So we have an interest in its relationship to addiction, obesity, chronic pain, but also it's very, very common for people as they get older to develop depression and associated with degenerative diseases. And, you know, it was back in 1990 that we all thought we'd cured depression with the launch of Prozac, a serotonin-specific reuptake inhibitor. But as this article article from 2018 in New Scientist discussed, nobody can actually agree about this antidepressant. And there's been a lot of concern that whilst for some people it undoubtedly does give them some benefits, for many people their mood disorder still remains a problem. And this is where in 2000 a very, very exciting development was reported in the literature. And unlike any of the other drugs that had been used to treat depression, this was a drug that was well known. It had been a club drug, it had been a drug of abuse, it was also an anesthetic. And it was found that at really low doses, this drug ketamine, we call it an NMDA antagonist, was not only antidepressant, but it was a rapid acting antidepressant. And why this is so important is because conventional antidepressants take about 6 weeks before people start to feel better. And that's really challenging for anybody. So you get diagnosed with a disease, you're told that, you know, you have a mood disorder, and you're given a pill that probably gives you some side effects, but it's 6 weeks before your mood starts to elevate. Ketamine is completely different. When it's given to patients, you can see this is the time scale of the ketamine effect. So this black line declines with as mood improves. And you can see that this is occurring very, very quickly. So this occurs over hours. And the interesting thing is that after a single dose of ketamine, patients were remaining well for many days. So this is over the sort of hours and days timescale. And this other graph just tells you a little bit about the acute effects of the drug. So people do experience the dissociative effects of ketamine, as you would expect. Even at the low doses, it's used as an antidepressant, but it's really short-lived. And the alignment is that this passes, and then the antidepressant effects seem to come in later. And this has been a really exciting but quite challenging finding, because it occurs clinically, and then we've got to start to try to unpack why this is happening. The other big finding is that it's very, very good at stopping suicidal thoughts. So it's seen as a sort of a potential anti-suicide drug, something we've never had before, because if you think about it, a drug that takes 6 weeks to work is really not going to be effective in somebody who is in acute phase where they're feeling very suicidal. Here we go. At 40 minutes, suicidal ideation, as we describe it, has dropped dramatically, suggesting that this is able to rapidly change somebody's mood. And the exciting thing for us is it's not just ketamine. It turns out that actually there's a number of drugs of abuse which appear to have these rapid acting antidepressant effects. You may have seen the BBC programme yesterday about psilocybin, the psilocybin trial. And the kind of common feature of these drugs is they are all drugs of abuse. They're all associated with a change in someone's perception acutely, be that MDMA, also well known as ecstasy, LSD, psilocybin, which comes from magic mushrooms, and another rather interesting compound, which is called scopolamine. And scopolamine is perhaps in some ways best known for its role in facilitating crime. So it turns out that 70% of people who've been robbed in Colombia have been exposed to scopolamine. Its nickname is devil's breath. But at low doses, it again has these rapid acting antidepressant effects. And you can see that the field has in general got very excited by this. So these are trials that are taking place against time. So you can see now we have a number of trials looking at the potential to use these drugs to treat depression. But of course, there are problems. These are drugs of abuse, and as it stands, you know, people have concerns about what may happen exposing people to these, and that some of that may be real, some of it may be perceptual, but it's something we need to consider. There are side effects. It's a potent, certainly if you watch the program, you could see these people were having very powerful psychological experiences. And that will put some people off. And particularly, we've noticed that there is quite a stigma, because these are drugs of abuse. There's quite a stigma associated with being treated with these. And that's an issue, because we want to be able to use this to help a wide population of patients. So this is where I come in. So I'm a basic scientist, and my interests are in how we might develop better rapid-acting antidepressants. So what we want to do is we want to, very similar to drugs that were discovered in the 60s, by chance, serendipity, led us to the SSRIs. These are drugs that are now the most commonly used antidepressants. And it was the science of understanding these tricyclic antidepressants, as they were called, that led us to make them better by developing the SSRI. So the question is, can we do the same with ketamine and, of course, these other drugs of abuse? Can we work out which bits of ketamine are associated with the beneficial effects? And could we separate those in some way from some of these side effects and make new drugs? Now, studies in patients are complex practically, but also complex ethically, and sometimes we need to move to a different setup. And of course, then we have the challenge that rats or mice, which is the most common laboratory species for developing new drugs, they're not really able to lie on a couch and tell us about their emotional experiences. So how do we undertake these types of studies? And that's been something that I've tried to really move forward in my field. And so we're interested in studying these emotional behaviors in these non-human species. And it has been a challenge, but this is what we've done. So we've actually gone to the human literature. We've tried to understand what is it that a patient with depression is experiencing, and is there a particular behavior that we could model in our animals? And the one thing that we've learned is that in depression, people attribute less value to positive experiences, and we refer to this as a negative bias. These biases affect mood because our mood is a product of our memories, and our memories then impact on our future expectations. And there's a neuropsychological kind of a basis to depression that these affective biases fit into. And we've been able to develop an animal model that's really quite simple, but actually does tap into exactly these same processes, we think, between humans and animals. Affective biases associated with memories. And the way we do this is a task called the affective bias test. It's a task that actually rats quite enjoy doing because it's a foraging task, and they have to dig in substrates in bowls to find rewards. But what we do is we train them, but that particular substrates predict reward and another substrate doesn't have any rewards. They have to learn that. We keep the value of the reward the same, and then we can bias their decision making by changing their affective state. And we've shown that in depression, we can model what's seen in people. They attribute less value to their reward memory than a control animal would. So this is what happens in depression. And then we've shown that ketamine at 1 hour can prevent that from happening. And most excitingly, we found 24 hours later, we've actually flipped the emotional association with that memory. It's actually become positive. So ketamine is doing something very clever in the brain and enabling that switch to happen, where a negatively biased memory is then becoming positively biased. Now, this is only the start. We now have the animal model, and we've got replication of this with other examples of these drugs, and we're able to now start to explore novel compounds, and we're working with a number of different chemists and looking at developing these new compounds. So what we've achieved so far is we've identified a neuropsychological mechanism that could explain the effects of rapid-acting antidepressants, and we've been able to use this in an animal model, replicating a major component of what's seen in people. We have the potential to now identify better treatments, and hopefully these will be able to have rapid and hopefully sustained effects. So in fact, we've got some very early data on a novel compound that we think is able to achieve both parts of the antidepressant effect. So mimicking ketamine and the SSRI. So that's quite an exciting early development. And therefore we can make better treatments. But also we've started to try to model in animals the relationship that might exist between changing the brain state using pharmacology. So that's what happens when you give, say, psilocybin, and then how that might interact with psychological treatments. And how does that then, can we potentiate this effect? Can we make this even greater or make this even longer lasting? And the other thing we can also do is something a little bit more fun, which is we can actually ask some questions that exist out there in the literature. So I've just got a couple of examples I want to finish with in how, because we now have an objective way of measuring emotions in animals, we're not using anthropomorphic interpretations. We're actually using a really clear, objective and validated measure. So one of the questions that's always bugged me is exercise antidepressant? Everyone sort of thinks that exercise should be antidepressant, but there's been a lot of controversy in the studies. And so one of the studies that showed it wasn't antidepressant was that people were given a gym subscription and a personal trainer. So what we said was, okay, if we put rats into a treadmill, aka a gym and a personal trainer, and then we compare that to experiences that they learn when they're in a home cage, do they show an affective bias? I.e., do they find this a more positive experience than this? And it turns out it's rather interesting. So actually, most rats are very lazy. Most rats actually don't enjoy what we would call forced exercise. I personally feel along those lines that the personal trainer might be a bit much for me. And the rats thought the same. So these rats didn't like the exercise, but some did. And so, in fact, some animals found this quite positive. So it did have an antidepressant effect. But interestingly, it's across the population. So we're suggesting that perhaps exercise, well, certainly this form of exercise, it could be antidepressant, but only if you like it. And then the other study we've also been doing, which aligns much more to the animal welfare side, is that there's one of these things that's been around for quite a long time, is that rats like being tickled by people. And they give out these vocalizations, and we think that the amount they vocalize may be a reflection of laughter. And so we did the same sort of experiment, and we tickled them one day and we didn't tickle them the other day. And we said, do you find the tickling day more positive? And we got a hugely significant correlation where actually the rats that are the most likely to vocalize were also the rats that had the most positive experience. But not everybody likes to be tickled, which of course is very similar to humans. Interestingly, nobody disliked being tickled, but they didn't all like being tickled. And this correlation with the vocalizations also suggests that when they are emitting these 50 kilohertz calls, these are related to their affective state. So that's just a really quick synopsis really of some of the sort of areas that we've been working on. And of course, I need to thank all the people who fund the work that we do and those that take part in the studies. So thank you very much for your attention. Welcome any questions.
Thank you so much, Emma. That was really interesting. And I can definitely empathise with those rats. I don't think I would like forced exercise either. I'm sure that our audiences have got some questions for you. So let's bring some of those up. A question from Lucas is, why are there concerns calling them drugs of abuse, given that there were already extensive research into psychedelics for mental health in the 1960s?
It's a really good point, isn't it? And actually, it's social, probably. It's the fact that back in the 70s, there was a real push to really make people quite fearful of psychedelic drugs, that the stories that it was going to make people jump off buildings or have bad trips. And the important thing, I think, is that we need to provide reassurance to people that these drugs are going to be used in a very controlled setting with somebody who's going to be with you all the time to provide reassurance. But I think also, just as a drug of abuse, they don't appeal to everybody. I think the prospect of having a very powerful, emotional, hallucinogenic experience will put off a number of people from using the treatment. And in fact, we know that's been happening with the ketamine clinics, that there are some people who are very fearful and they won't go forward for treatment. And so if we can find treatments that they may not be quite as effective, but if they can at least tap into some of the mechanisms, then we might have something that we can use in the interim. I think the other thing is going to be the cost implications. We know how challenging it is at the moment. People being given SSRI should be getting psychotherapy. We know that that makes the drug work better, but they don't. Most people are going to their GP, getting a prescription, and that's it. So I think it's a cost factor as well. We do want to try and find ways to make this available to as many people as possible.
Mm-hmm. Yeah, thank you for that. So we've got another question here from Albert. Does it look like you'd have to take, for example, ketamine long-term to keep depression at bay? Would that build up in the system and then have negative or addictive side effects?
Fantastic question. So actually what's really interesting is that you don't need to give this drug every day. So the most frequently that's been tested is twice a week. And that does reduce the risks of any associated addiction. But equally, we are aware that some people have said that they've experienced their depression getting better. Going to a clinic can be a challenge when there's a drug available on the street. And so there is a concern that people may turn to an illicit source, and then they may take higher doses, or more frequently, and that may be a problem. Mm. What people think at the moment is that you need to give these drugs. So ketamine is about once a week initially, and then but it's the combination with psychotherapy. I think nobody is seeing this as a cure pill. What this looks like is it's a way of sort of going for a reset. It restarts the brain's emotional processing mechanisms. It allows for more positive emotional processing, but you then have to deal with the other factors that were driving the depression. So if people make no changes to the factors that are driving their depression, that maybe they can't, or that they haven't undergone sufficient sort of psychological treatments alongside, then you will see a return of the depression. And there's a mixed bag at the moment. So it's working for some people where they go less and less and less, bigger distance between the ketamine, they get better, and then they stay well for a long time. Other people are needing to still have it, say, once a month. But even so, it's massively improving for them, their experience of their mood disorder.
I mean, we've learned from tonight that the brain is incredibly complicated, so it's not surprising that just taking a pill is not the only solution to something like depression.
Yeah.
So I think we've got another question to bring that up. We've got one from Isabel. What do you think is the most challenging barrier to proceed with these types of treatment? People willing to take it, pharma companies, policies suited for it?
Great question. So policy has been a big problem for all of us in research, from basic science through to the clinical trials. 2 years to get a license to give people psilocybin, many barriers. I mean, to the point that actually it's only recently that we've even been able to start to even try and do this research. So I think policy has been a problem. I think that is changing. So we're now looking at, I think there are actually a lot of people want to take part in the trials, but that is a subpopulation. So I don't think we have an issue with the trials. Pharma companies, we've got a fantastic company who has set up their entire kind of ethos around not just the drug, but also the drug and the digital support and the psychotherapy and that whole kind of program. So I think we have got business interest, which is critical to supporting these costly experiments. I think the biggest barrier is understanding how they work, because actually, we discovered the first antidepressants in the 1950s, 60s, and we still don't know how they work. So we have made refinements and we have some understanding, but I can't tell you why an antidepressant makes you better and not somebody else. That's still something. So I think the barrier is really investment in understanding the causes of depression. And personally, not my field, but I'd like to see us look much more at the societal causes of depression. It's a Western disease. It increases the greater urbanization, the more capitalist the country is, these things are associated, and I'd like to know more about why, because I think in many ways prevention is the most important thing we can do. And so understanding mechanisms, understanding how we adapt our society such that we really put mental health high up on our agenda in terms of recognizing success rather than perhaps always driven by money.
Definitely. I think that is so important. And I hope that understanding the mechanism a bit more as well will help to get rid of that stigma. I think we've got time for one last question before we wrap things up. So, question from Dan. On the ketamine suicidal feelings point, is there anywhere in the world that uses ketamine to prevent suicide? Do you see a time that we see this on the NHS or even in certain suicide spots?
So yes, so there's a company, Janssen, who developed a version of ketamine called S-ketamine, and there's a nasal spray, and that has a license for the treatment of treatment-resistant depression. And I think it's currently in investigation. I expect it will come to the market as a sort of A&E type treatment. So people will come into hospital because they're suicidal. They'll be given a dose of ketamine in the hospital setting. It's not a drug that you give to people to take away, so it would be in a hospital setting, but it should make them stable for several, certainly for the period of 24 hours till you can get mental health services involved. And I think that's going to be really important. Bristol is setting up its first clinic. It is a private clinic, but it is based around ketamine treatment. And there's also obviously the Oxford ketamine clinic's been running for some years. Will it come through on the NHS? It's an interesting one because ketamine is a really cheap drug that's been used a lot for lots of reasons. And at the moment, the NHS isn't licensing it, but I think that will change. I think as more of the evidence builds, I can see us using it. Possibly not the expensive version of S-ketamine, but I think it will definitely.
Amazing. So I think that's all we've got time for, but thank you so much, Emma. We've got loads of questions for you, but I'm sorry if we didn't get to your question. We're going to have to wrap things up now. So let's first take a look at our audience answers to the second trivia question, which was, what is the total information content of the human brain in bits? So Megan says 10 million bits. Nabina says 15 million. Joe says 100 million. Rose says 20 trillion. Wow, it's a bit higher from Rose. I think that's maybe all we've got, but I can tell you what the real answer is. So there's some good attempts, but I can reveal that the answer is in fact somewhere around 2.6 times 10 to the 42 bits. So I don't know how to say this in words, but I think we'll have a comment in the chat function which spells that out in words. But to give you an idea in real terms of how much that is, if your brain could be expressed as a song that could be downloaded, that song would last for 50,000 trillion years. So thank you, Danielle George, for sharing that amazing fact with us last night. Before we leave tonight, I just want to give a final thank you to all of our wonderful speakers for giving such interesting talks. It's been a real enjoyable event for me as well as the audience, I'm sure. And I'd like to thank the entire Pint of Science team for helping to make this event happen, as they're all currently working hard behind the scenes to make sure that this show runs as smoothly as possible. So a big thank you to them. And finally, I also want to thank our audience for taking part. Your contribution has really made the show, so thank you for all of your questions and comments. It's been great to be able to interact with all of you, even in a virtual world. So I hope that you enjoyed the show tonight and that you learned something new. Cheers and good night.